Imagine a molecule so small yet so mighty that it dictates whether you'll rock blue eyes or brown, decide if your morning coffee is bliss or disaster, and even hold the secrets to curing once incurable diseases. That's DNA, life's blueprint found in every cell of our bodies. DNA contains genes, the instructions for building and maintaining organisms. These genes determine traits like eye color and disease susceptibility. Sometimes changes called genetic mutations occur, altering these instructions. They can lead to genetic disorders or, in some cases, offer advantages in specific environments. In medicine, DNA is crucial. Antibiotics, for example, disrupt bacterial DNA to fight infections, but bacteria can develop resistance through mutations. Viruses use genetic manipulation to invade and replicate within cells. Meanwhile, genetic engineering like CRISPR-Cas9 allows precise editing of DNA, promising treatments for genetic diseases and advancement in agriculture and biotechnology. DNA is more than a molecule. It's the key to understanding and transforming life. What if it holds the secrets to conquering diseases, unraveling mysteries, and pushing the boundaries of what's possible? So if DNA is the answer, does that make genetics the ultimate game of hide and seek? Hey everyone, I'm Ashley of Meet the Gen Z, and I'm thrilled to be your guide on this daring, new, and amazing adventure. Here at Meet the Gen Z, we're on a mission to mutate your understanding, where we substitute old knowledge with new insights, clear out misconceptions, and infuse fascinating information. Oh, and did you hear about the scientists who figured out how to make a DNA laugh? They cracked the code! Today, we're diving into the fascinating world of the other aspects of nucleic acids and protein synthesis. By the end of our adventure, you'll achieve the following learning outcomes. 1. Define mutation. Understand how genetic changes can lead to variation in traits and diseases. 2. Describe hereditary diseases and their impact on biological functions. 3. Recognize the importance of antibiotics in combating bacterial infections. 4. Explain the mechanisms by which antibiotics inhibit bacterial growth. 5th, illustrate the process of viral infections and replicate within host cells. 6. Outline the steps involved in recombinant DNA technology and its applications. 7. Describe how transgenic organisms are created and their implications. 8. Discuss the impact of genetic engineering on advancement in medicine. And lastly, understand the ethical considerations surrounding genetic engineering practices. And let's not forget our roadmap through this scientific expedition. We'll start with genetic mutations, where we'll discover the complexities of DNA's twists. Next, we'll explore antibiotics, tiny warriors in the ongoing battle against bacterial threats. Following that, we'll shed light on viruses, masters of genetic manipulation and adaptation. And finally, we'll delve into recombinant DNA technology, where we'll manipulate genetic building blocks to forge new frontiers in scientific discoveries. Now, what are you waiting for? Let's mutate our understanding as we dive into our first stop, genetic mutations. So let's kick it off with a fun pre-discussion activity. Can you guess who these characters are? If you answered Teenage Mutant Ninja Turtles, Wolverine, and the Powerpuff Girls, then yes, you are correct. Now my question for all of you is, what do they all have in common? I mean, what are their similarities? Exactly. All of those characters have undergone some sort of mutation in their structure. Unlike the mutations that give superheroes their powers in movies, mutations are a real-world occurrence that typically do not result in extraordinary abilities. In our previous video, you learned that the base pairing mechanism provides an almost perfect way to copy a DNA molecule during what process again? DNA replication it is. The key word here is almost. No machine, not even the copying mechanism of DNA replication, is totally without error. And do you guys have an idea of what this error is called? Correct again. A mutation is a change in the sequence of bases in DNA. These mutations occur in two ways, spontaneously or induced. Well, these spontaneous mutations result because of errors in base pairing during DNA replication. However, these errors are very rare. It has been estimated that, on average, one error occurs for every 10 to the power of 10 bases. Now, can any of you tell me why mutations are very rare based on what you've learned previously in the last meeting? Yes, you got it. It is because of DNA polymerase and some other enzymes that have repair functions. Again, mutations are not only spontaneous. These could also be produced by what we call mutagens. Mutagens include ultraviolet light, such as sunlight, and various high-energy radiations like X-rays, cosmic rays, radioactivity, and a variety of chemicals. So if mutagens react with nucleotide base pairs by either converting them to other bases or to other structures, the good thing is that there are repair enzymes that minimize the effects of mutagens. Additionally, the overall effects of mutagens depend on their amount and the duration of exposure. Now here's a fascinating tidbit about mutations. How many of you are lactose intolerant? About 12,000 years ago, a single human developed a genetic tweak that enabled them to digest milk from cows, something previously unheard of. Now, this mutation from lactose intolerance to lactose tolerance is now so widespread that entire industries are built around dairy products. So mutations occur through substitution, insertion, and deletion of bases. So the most common type of mutation is substitution, which involves the substitution of one base by another, changing one codon in mRNA. Now take a look at this picture. What have you noticed? Bingo! A base in the sequence got substituted. This may or may not alter the amino acid residue specified by that codon because of the degeneracy of the genetic code. An insertion mutation, on the other hand, involves the insertion of a base into a normal sequence. Now take a look at this picture. Can anyone tell me what happened? Yes, some bases were added. Lastly, the deletion mutation involves the insertion of a base into the normal sequence. Now again, take a look at this picture, and can anyone describe the picture? A base was removed from the sequence. Now, since protein coding DNA is divided into codons, 3 bases long, insertions and deletions can alter a gene so that its message is no longer correctly parsed. These changes are called frameshifts. For example, consider the sentence, "The fat cat sat." Now, each word represents a codon. If we delete the first letter and parse the sentence in the same way, it doesn't make sense. In frameshifts, a similar error occurs at the DNA level, causing the codons to be parsed incorrectly. This usually generates truncated proteins that are as useless as just read it here. It is uninformative, yes. Moving on, mutations that have negative biological consequences fall into 2 categories: mutations in somatic cells and mutation in germ cells. Now let's talk about the former first. Mutations in these cells affect the biological functioning of an individual organism. The individual may become sick or even die. An example of a consequence of this mutation is cancer. A mutagen that causes cancer is called a carcinogen, and this is the most common mutagen. Moreover, mutations in egg and sperm cells have much greater and far-reaching significance since they can be passed on to subsequent generations. Now, these mutations are what we call genetic defects or hereditary diseases. Now, let's delve into some examples to understand these impacts more deeply. Albinism. Imagine life without the protective pigment melanin in your skin and your eyes. People with albinism face increased risk of sunburn and skin cancer due to their lack of melanin. Cystic fibrosis. This genetic disorder affects the lungs and the digestive system due to inadequate production of lipase, leading to the excessive mucus production and severe breathing and digestive difficulties. Diabetes mellitus. A malfunction in insulin production or function results in poor glucose metabolism, leading to high blood sugar levels that can cause atherosclerosis, vision problems, and circulation issues in the extremities. Galactosemia. Individuals with galactosemia cannot metabolize galactose properly due to a defective enzyme, leading to its accumulation in cells and causing complications such as cataracts and cognitive impairments. Hemophilia. This disorder results from a deficiency in blood clotting factors, making it challenging for blood to clot properly and causing prolonged bleeding and internal hemorrhaging. Phenylketonuria, or PKU. In PKU, the body lacks an enzyme needed to break down phenylalanine, leading to its toxic accumulation and resulting in intellectual disabilities and neurological problems. Sickle cell anemia. This condition affects hemoglobin, the protein in red blood cells responsible for carrying oxygen. Abnormal sickle-shaped cells can block blood vessels, causing pain, anemia, and organ damage. And lastly, we have Tay-Sachs. Tay-Sachs disease is a rare genetic disorder where a missing enzyme causes harmful substances to accumulate in the brain and nerve cells, leading to progressive neurological decline and loss of motor skills. And there you have it, guys. That's the genius wrap-up on genetic mutations. Now, here's a challenge for you. Think of one beneficial mutation that you wish humans could develop and why. Get creative, whether it's the ability to see in the dark, resist the common cold, let your imagination run wild. Share your ideas in the comments below and let's spark some genetic creativity. Now that we've delved into the fascinating world of genetic mutations and seen how these changes in DNA can lead to significant biological differences, let's shift our focus to another crucial topic in our study of nucleic acids and protein synthesis. Imagine just for a moment a world where something as common as a strep throat couldn't be treated, where even a simple cut or blister could become life-threatening, regardless of where you live or how advanced your healthcare system is. This scenario might sound like science fiction, but it's a very real concern if antibiotics lose their effectiveness against bacterial infections. So antibiotics are medicines that help stop infections caused by bacteria. And as a clue to understand the word antibiotics, you can see it has the words anti and bio, meaning against life. Now, this may sound scary, but its purpose is specific for those bacteria only. Now, there are many different types of antibiotics. Can you guess why we have different types of them? If you guess that it's because they target different types of bacteria in different ways, then yes, you are correct. As our main topic is about nucleic acids, it's given that these antibiotics can act on the DNA, RNA, and or other structures that make up the bacteria. In principle, there are three main antibiotic targets in the bacteria. First, the cell wall or membranes that surrounds the bacterial cell. Second, the machineries that make the nucleic acids, DNA, and RNA. And lastly, the machinery that produce proteins, the ribosomes and associated proteins. Now that you have a gist of how antibiotics work, let's dive into the nitty-gritty and uncover the antibiotactics these bacterial busters use in their mechanisms of inhibition. First up, we have actinomycin. This one is like a strict librarian binding to DNA and preventing it from being a template for both DNA and RNA synthesis. It basically stops the bacterial story from being written. Next is chloramphenicol. Imagine it as a chef who interrupts the kitchen, inhibiting peptide bond formation by interfering with peptidyl transferase. No peptide bonds means no proteins, and no proteins means no bacterial growth. Then we have erythromycin. This antibiotic binds to the large subunit of the ribosome and prevents its movement along the mRNA, like putting a roadblock in the path of a delivery truck, halting its production line. Penicillin, it's another classic. It inhibits cell wall formation by blocking the formation of a needed enzyme. Think of it as preventing the bricks from being laid in a wall, causing the bacteria to lose its protective barrier. Now moving on to paromycin, this one binds to the A site of the ribosome, becomes attached to the growing polypeptide and causes premature termination. It's like cutting off a story mid-sentence, leaving the bacteria with incomplete proteins. Now streptomycin works by inhibiting initiation and causing the incorrect reading of mRNA by binding to rRNA. It's like messing up the blueprint, leading to faulty bacterial construction. And last but not the least, we have tetracycline. This antibiotic binds to rRNA and prevents the binding of tRNA to mRNA. Imagine it as a gatekeeper, stopping the tRNA from delivering its amino acid cargo to the mRNA, effectively halting protein synthesis. Now that we've covered how antibiotics work, let's talk about antibiotic resistance. Antibiotic targets the cell wall, DNA and RNA machineries, and protein-producing machinery of bacteria. But bacteria can mutate, changing their genes to survive these attacks. Now, when bacteria encounter antibiotics and survive, they can pass on their resistant genes to their descendants. This is how antibiotic resistance strains develop. Gene mutations in bacteria can enable them to ignore the antibiotic, block the antibiotic, expel the antibiotic, alter the antibiotic's action, destroy the antibiotic, and divert the antibiotic to different targets. Now, these mutations and the widespread use of these antibiotics have led to resistant strains of common bacterial infections, especially in hospitals. Now, the key here is that bacteria undergo gene mutations, which can make them resistant to antibiotics. These mutations are then passed on to creating a population of resistant bacteria. Now, to prevent this, we need new antibiotics, proper use of existing ones, and continued research into bacterial behavior and resistance. This is all for our topic about antibiotics. Now, it's time to put your knowledge under the microscope and see if you can cure these questions. First, what do you call medicines that help stop infections caused by bacteria? Yes, the correct answer is antibiotics. If you got that right, give yourself a pat on the back. Now, what are 3 things antibiotics target on the bacteria? Yes, antibiotics target three main areas in the bacteria. They interfere with the cell wall or the membrane surrounding the bacterial cell, disrupt the machinery responsible for making nucleic acids like DNA and RNA, and inhibit the machinery that produces proteins. Now lastly, why do bacteria develop resistance against antibiotics? Yes, because bacteria develop resistance through genetic mutations and by sharing resistant traits. Overuse and misuse of antibiotics accelerate this process, creating strains that are harder to treat. And that is all for antibiotics, everyone. Remember, knowledge is your antibiotic against ignorance. Alright, everyone, let's take a moment for a quick brain break. Now, close your eyes and take a deep breath in and exhale slowly. Again, deep breath in and exhale slowly. You feel refreshed? Now let's stretch out those brains with a fun fact challenge. I'll share a fascinating fact and you think about how cool it is. Ready? Fact. Did you know, despite our differences, we're all 99.5 genetically similar, and only about 0.5 of the 3 billion base pairs in the human genome vary between individuals, contributing to our uniqueness. Amazing, right? Now, let's take another deep breath in and exhale. And let's get back to our learning journey. Alright, hope everyone's feeling recharged after that brain break. And now let's zoom in on the mischief maker behind the COVID-19 pandemic, viruses. From its unexpected global impact to the race for vaccines, viruses like COVID-19 have shown us just how powerful these tiny entities can be. Now, viruses are infectious, parasitic small particles containing either DNA or RNA, but not both. They are everywhere in our environment, from the food we eat to the air we breathe. We take in millions of viruses every second. Now, this may sound scary, but actually it's normal. Viruses are responsible for a wide range of diseases such as influenza, leukemia, smallpox, and hepatitis. Now, I have a question for you. Do you think viruses can replicate on their own? If you said they don't, then yes, you are correct. Viruses need a host like we need our morning coffee. They cannot reproduce or synthesize proteins because they lack amino acids, nucleotides, or even enzymes necessary for replication, transcription, and translation. Essentially, they're considered non-living organisms. And now that we've mentioned hosts, can you tell me what this picture shows us? Yes, what you are seeing here is a virus infecting a host cell. An infection of a host cell by a virus occurs when the virus sticks to a host cell and then its cell membrane is dissolved by an enzyme present in the protein overcoat of the virus. This is then followed by the entry of DNA or RNA of the virus into the host cell. Now, there is high specificity in this process as only certain types of host and host cells are invaded by any virus. For example, HIV, which is responsible for AIDS, infects only the T cells of the immune system. The infected person is then vulnerable to opportunistic diseases. Now that I've explained how viruses infect a host cell, can you speculate on how they reproduce? Voila! And here's how viruses reproduce. The viral genome takes over the host cell machinery, making it replicate the viral genome and reproduce proteins. These proteins then assemble new viral particles with protective capsids, enabling viruses to multiply and spread within their host. However, the reproduction of the virus actually differs on whether the virus contains DNA or RNA. So, we have two types of viruses, DNA viruses and RNA viruses. An RNA virus is a virus that has an RNA as its genetic material, and, of course, a DNA virus is a virus that has DNA as its genetic material. The DNA of DNA viruses is transcribed into mRNA by the host cell. The viral mRNA is then translated into viral proteins. Now, these viral proteins then assemble to form new viral particles. Basically, the DNA virus replicates in the same manner that the host replicates on its own. RNA viruses, or also known as retroviruses, on the other hand, replicates in three ways. 1. Some RNA viruses uses their genome directly as mRNA. After infecting a cell, their RNA is translated directly into a new viral protein. Other RNA viruses carry enzymes that help their RNA genome act as a template for the host cell to reproduce viral mRNA. Now, third, other retroviruses like HIV uses reverse transcriptase to convert their RNA into DNA, and this viral DNA forms a double-stranded molecule that is transcribed and translated by the host cell, allowing new virus particles to be produced. This process, known as reverse transcription, challenges the central dogma of molecular biology by reversing the usual flow of genetic information from DNA to RNA. Wow! That was a whirlwind tour through viral replication, right? Now let's slow things down a bit and take a closer look at the general flow of how viruses replicate. Take a look at this diagram. And there you go. We've explored the mysteries of viral replication and taken a closer look at how these tiny invaders multiply. Remember, viruses may be small, but they have a huge impact on our world. Now get ready for our next adventure into genetic engineering. Now, just as engineering transforms building, today we'll explore how it transforms the very building blocks of life through recombinant DNA technology. Now, our last lesson is the combination or the alteration of DNA, also formally known as genetic engineering or recombinant DNA technology. And this is done with the objective of having an organism produce a desired polypeptide. More specifically, recombinant DNA technology is a technology used for producing artificial DNA through the combination of different genetic materials or DNA from different sources. Inserting the desired gene into the genome of the host is not as easy as it sounds. It involves a selection of the desired gene for administration into the host, followed by a selection of perfect vector with which the gene has to be integrated and forming recombinant DNA. Thus, the recombinant DNA has to be introduced into the host, and at last, it has to be maintained in the host and carried forward to the offspring. But how exactly do scientists achieve this? Let's break it down. Step 1. Selection of a vector genome. Choosing the right vector is crucial. E. coli or Escherichia coli is most often used because they contain two DNA forms, one in its single chromosome, and others are the plasmids, which are numerous circular double-stranded DNA. Their plasmids can easily be isolated, altered, and then reintroduced into the E. coli. Step 2. Isolation of genetic material. The next step in recombinant DNA technology is to isolate the desired DNA in its pure form, free from other macromolecules. This involves enzymes such as lysozyme, cellulase, chitinase, ribonuclease, protease, all the A's that you can think of. Yes. Step 3. Cutting the gene at the recognition site. The restriction enzymes play a major role in determining the location at which the desired gene is inserted into the vector genome. These reactions are called restriction enzyme digestion. Think of these enzymes as tiny molecular scissors. Step 4. Amplifying the gene copies through polymerase chain reaction or PCR. It is a process to amplify a single copy of DNA into thousands, millions of copies once the proper gene of interest has been cut using restriction enzymes. PCR is like making thousands of copies of your favorite song but with DNA. Step 5. Ligation of DNA molecules. In this step of ligation, the joining of two pieces, a cut fragment of DNA and the vector, together with the help of the enzyme DNA ligase. DNA ligase is the glue that holds our genetic masterpiece together. Last, step 6 is the insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into a recipient host cell. This process is termed as transformation. Once the recombinant DNA is inserted into the host cell, it gets multiplied and expressed in the form of the manufactured protein under optimal conditions. Amazing, right? Now that I've unleashed the secrets to recombinant DNA technology, can anyone share some real-world examples of its handiwork? Come on, don't be shy and let's see those comments buzzing. So moving on, two notable products of the recombinant DNA technology include human insulin, crucial for managing diabetes, and growth hormones used to treat growth disorder. Another significant example is interferon, which boosts the immune system against infections and cancer. However, these advancements also raise ethical concerns about their impact on human animals and the environment. It's a double-edged sword. Genetic engineering raises ethical questions impacting humans and the environment. Concerns including unintended consequences from modifying genetic codes due to complex gene interactions, necessitating rigorous regulation. First, environmental impact. Genetically modified crops and animals can interact with wild species, leading to unintended effects like superbees and ecosystems disruption, altering biodiversity and ecosystem stability. Second, informed consent and fairness. Obtaining consent for genetic modifications, especially those affecting future generations, is complex. Ensuring parents understand risk and benefits is crucial, with fairness issues arising from unequal access to genetic technologies, potentially exacerbating health disparities. Third, animal wealth. Genetic modification in animals may cause unforeseen physical and behavioral changes, posing ethical questions about animal welfare and rights. Now, as we venture deeper into the world of genetic engineering, let's remember to keep our ethical compass aligned. Just like the double helix of DNA, understanding these ethical twists and turns ensures our innovations spin in the right direction, benefiting society and the environment responsibly. Alright guys, now let's wrap up our crash course on mutations, antibiotics, viruses, and recombinant DNA technology. We have covered a lot, so here's a quick review to lock it all in. Mutations. Mutations are changes in the DNA sequence caused by errors during replication or exposure to mutagens like UV light or chemicals. Now these changes can be beneficial, harmful, or even neutral, influencing genetic diversity and evolution. Antibiotics. Antibiotics are medicines that target bacterial infections by disrupting cell walls, nucleic acids, or protein synthesis. However, overuse and misuse can lead to antibiotic resistance, where the bacteria evolve to resist these drugs, posing a serious global health threat. Third, viruses. Viruses are infectious agents that require host cells to replicate. They contain either DNA or RNA and infect specific cells such as immune cells in HIV or AIDS. Viruses hijack cellular machinery to produce more viruses, spreading infection. Fourth, recombinant DNA technology. This cutting-edge technology combines DNA from different sources to create new genetic sequences. It's used to produce essential medications like insulin and vaccines. However, it does raise ethical concerns about safety, consent, and environmental impact, highlighting the complex intersection of science and society. And there you have it, a whirlwind tour through some of the most fascinating and impactful aspects of genetic research and biotechnology. And now that we've refreshed your understanding, it's time for a DNA test. Now to test your knowledge, I'll be asking a few questions based on what we've covered, and drop your answers in the comments below and let's see how much you've learned. 1. What are mutations and how do they contribute to genetic diversity? 2. Why is antibiotic resistance a significant concern in healthcare today? Third, how do viruses replicate inside host cells and what makes them challenging to treat? Lastly, how is recombinant DNA technology used to create important medications like insulin? Remember to be specific in your answers and feel free to discuss with the others in the comments below. Now let's test your genetic knowledge. As we conclude our journey through mutations, antibiotics, viruses, and recombinant DNA technology, we've witnessed the fascinating web woven by these tiny molecules. DNA isn't just a blueprint, it's the language of life itself, shaping our past, our present, and our future. From genetic mutation driving evolution to antibiotics protecting our health, and from viruses testing our resilience to genetic engineering revolutionizing medicine, DNA molecules are pivotal in understanding and changing our world. So as we marvel at the wonders of genetic science, let's ponder, how will our growing knowledge of DNA shape the next chapters of human progress? I hope you enjoyed diving into these topics as much as I have. Keep exploring, keep learning, and most importantly, keep asking questions. Together, we are unraveling the mysteries of life itself. Until next time, and stay curious.